Hello and welcome to episode 15 of this lecture series, and today's perfume is Eden or Eden, a 1994 composition by one of the greatest perfumers, in my opinion, of the 20th century, Jean Guichard, who worked for Givaudan, a major fragrance company. And Eden is, first of all, is an instance of perfect agreement between the color of the bottle, the shape, the smoothness of it, and the actual fragrance. The fragrance smells, when I first smelt it, I thought it was kind of creamy, heavy green, and it smelled a little bit like a wet woolen sweater. And I remember writing about it that who wants to smell like a wet wool sweater? And on reflection, with the years passing, and me smelling this thing again once in a while, the conclusion I came to was, I do. It smells fantastic. It's also very durable and very linear, meaning it doesn't really change with time. It's one of the absolute greats. You can probably find this on eBay, on various places, very cheap. So today's episode will be dedicated to the objections to the vibrational theory. So far I've given you a beautiful linear story with a happy end, everything works out, and I have documented all the different steps that we went through from the first idea in 1996 all the way to the experiments on flies, on humans, all the computational stuff and everything. I made it sound like basically it was self-evidently obvious that the vibrational theory must be right and the shape theory is wrong. In fact, that is not the case, because a lot of people, possibly a majority of people in the field of olfaction, do not believe that the vibrational theory is correct, and they believe that it must be shape. So in this lecture, I'd like to go through the major objections that people have come up with to the vibrational theory of smell, and my answers to them. So the first objection is, well, we all know it must be shape. Well, that's a fairly easy one to get rid of, but it requires knowledge of fragrance chemistry that most biologists don't have. Most biologists do not, even those who are involved in smell research, do not necessarily talk to fragrance chemists. Fragrance chemists will tell you that fragrance molecules are discovered by accident. In fact, a very famous fragrance chemist who worked at Givaudan was well known for, when they made presentations of new molecules, for sitting at the back of the lecture hall, and when anybody claimed that they had done this rationally by design, he would, from the back, shout, "We got it by accident." He was the boss, so they couldn't kick him out. So biologists don't talk to fragrance chemists, and biologists are absolutely convinced that smell must work on shape, since everything else does. Now, this is simply not true. As I've explained, shape theories do not predict smell. They do not predict the fact that we can smell functional groups. They do not predict the isotope effects. They predict actually pretty much nothing. So that objection, it must be shape, is non-scientific. So a question I often get is, well, what about enantiomers? So let me explain what enantiomers are, first of all. Enantiomers are pairs of molecules that are like a mirror image of each other. Imagine that there's a mirror between my two hands. When I move my left hand, the right hand in the mirror does the same. And when I move the thumb or the index, they do the same thing. So many molecules exist as enantiomers. They have an asymmetric, what's called an asymmetric or a chiral center in the molecule, and that means that there is there really are two versions of the molecule, one corresponding to the left hand and one corresponding to the right hand. Now, why is this important? It's important because any vibration that this molecule undergoes will be mirrored by the same vibration in the other molecule. So that if you imagine that you have in a spectrometer, a normal laboratory spectrometer, a pure solution of this molecule, and you put it in a cuvette and you measure the vibrational spectrum, and then you remove the cuvette and you put a pure solution of this molecule and you measure the vibrational spectrum, they're going to be identical. So you might say, well, so what? Well, the fact is, some enantiomers smell different. But what's interesting is, if you look at the history of enantiomers smelling different, it took 30 years to prove that one famous pair of enantiomers, the two carvones, smelled respectively of mint and of caraway. It took 30 years to actually prove it. And this was considered the exception. Most enantiomer pairs smell identical. So the objection to vibrational theory is, these two have identical vibrational spectra. How can they smell different? And the answer is actually quite simple, but it's in two parts. Part one is this. When you have a spectroscope that is so small that it's roughly the cuvette, the place where you put the molecule, is the size of the molecule, which is what a receptor is, you will get what are called polarization effects. Namely, the electrons are forced to travel across the molecule in one direction, and it can only see, the current of electrons can only see certain groups, certain parts of the molecule, and not others. So for example, if this is a C double bond O, it will see the vibrations of this guy when it's oriented this way, but not this way. These are polarization effects. They're well known. They're experimentally well known. So in the most famous example of the two carvones, one that smells of mint, one of caraway, I showed years ago, in 93, 94, that if you added back a bit of C double bond O vibration to the mint one, it would smell of caraway. Even in a real spectroscope, if you take a crystal of a particular enantiomer, you will get a different spectrum depending on how it's oriented inside the spectrometer. So there's no real problem there. However, there's an absolutely major problem for shape theories in explaining any enantiomer pair that smell identical. And the reason is simple. Every single receptor will have some degree of asymmetry, of handedness. So it's impossible to imagine a receptor of any kind, any drug, that will bind equally well both enantiomers of a particular molecule, simply because one will fit better than the other. So that the vibrational theory has a ready explanation for the fact that some enantiomers smell different, but the shape theory has no explanation for the fact that most enantiomers smell the same. Another objection, which I find almost incomprehensible, to be honest, is people who say, well, isotope smell experiments, actually smell experiments in general, prove nothing. In other words, that it's always better to have either flies doing whatever they do, as we've done, flies choosing one smell over another, or cells in tissue culture responding to an odorant by something that you can measure with a microscope. But in the end, if you believe that the sensation of smell is meaningless, in other words, that you can learn nothing from smelling things, then why are you studying olfaction? I just don't understand why scientists, and it's this often, I've seen this happen often, people who are working in the field of smell don't smell things. They don't even know the smell of the molecules that they put on their animals and so on. They don't care. And this is very unusual because, for example, in the study of hearing, people take the hearing sensation very seriously. They will say to you, listen to this. Can you hear the fact that this or that happens? Or when they do a visual illusion, they can say, okay, what do you see? What is the color of that thing on the left? And you say green, and it's not green. So in other areas of sensation, people take the sensation itself very seriously. For some reason, in the case of smell, people have a hard time doing that. And I don't really understand why that is. And there have been various theories about it. But mostly I think it's just lack of familiarity. They believe that everybody smells things differently. But if that were true, the world wouldn't be what it is. There would be no perfumery, there would be no cooking, there would be none of the activities that are related to the beauty of smells would exist if we smelled things differently. Another line of criticism that people have come up with, which is at least more scientific than some of the ones I've mentioned so far, is to say, well, deuterium, as opposed to hydrogen, has other effects than just on vibrations. And that is true. It has other effects. But they come in two categories. It has very big effects on chemical reactions. If a deuterium, or a hydrogen atom needs to jump through a barrier, it will do so much more slowly when breaking one bond and making another than a hydrogen atom. So that's a big difference. But in smell, there are no chemical reactions. The odorant is not reacting with anything. It's just attaching and detaching. So there are no chemical reactions. More importantly, there are typically no hydrogen bonds made by the hydrogens in a molecule. A CH cannot make a hydrogen bond. So if you replace the hydrogens on carbons with deuterium, it will make no difference to hydrogen bonds, even though it is true the hydrogen bonds with deuterium are different. It doesn't apply to the situations that we've been dealing with because those hydrogens do not make hydrogen bonds. However, there is an instance in which deuterium is actually different to hydrogen, and that is a very small difference in the stickiness to an oily surface, to a hydrophobic surface. And we've seen that, for example, you see that when you do chromatography. You remember chromatography, we put isotopes into a column to purify them, and they go around and around and around 40 meters of column, and then they come out. Well, if you take the two musks and you put them in at the same time, they, after a good long time traveling inside the column, they come out separated with actually the deuterium coming out first and the hydrogen coming second. But it takes a 40-meter column to even achieve that kind of separation because the difference in stickiness to the walls of the column is absolutely tiny. Another objection which was quite prominent a few years ago was the fact that when people put olfactory receptors in non-olfactory cells that are in a dish, in a culture dish, so they put the receptor in, they put the rest of the machinery, and then they put odorants and they measure the response. The people who did this tested the response of these particular cells to a normal and a deuterium musk, just as we did. And they found no difference with those two odorants on those particular cells. And remember, these are receptors that have been taken out of their normal environment and put into a different cell that happens to be easy to measure cellular responses in. And so it is an absolute fact, the result, that those cells with that receptor, with those musks, did not respond differently to isotope. That article in a very well-known journal in which I've published several papers called Proceedings of the National Academy of Sciences of the US was accompanied by an editorial, by a comment, an editorial article, of which this is the front page, suggesting that the vibrational theory should be, quote, laid to rest on the basis of essentially one experiment. Now, first of all, there's no theory ever, really, that can be disproved by a single negative result. I can't think of any. And secondly, it turns out that that's not even true, that the cells don't respond differently to isotopes. There's more recent work using actual olfactory neurons that shows that a small proportion of these neurons respond in an all or nothing fashion to isotopes. So you have a very large majority of neurons don't respond differentially to isotopes, and this small population responds absolutely yes or no to isotopes. Those neurons, which are a minority, a small minority, 1%, are direct evidence for the vibrational theory. But when you read the paper, the way that it was written, it said, well, basically 99% of neurons don't care about vibrations. 1% do. Therefore, the vibrational theory is wrong. Suppose I say to you, pigs don't fly. Pigs don't fly. And then one pig comes flying by. And you say to me, well, okay, some pigs fly. What you've done is you've gone from there is no such thing as a flying pig to there is such a thing as a flying pig. It's a huge difference. And the way they wrote it was, those cells that do respond to isotopes are hypersensitive to small differences of hydrophobicity between octanal and deuterated octanal. Now, there is in physics and in physical chemistry no such thing as hypersensitivity. If you have a force of binding and it changes essentially 100% between a hydrogen and a deuterium, you have made a major discovery in physics. And that is not going to happen simply because the laws of physics go against it. So when people say the cells are hypersensitive, they're projecting a concept of hypersensitivity, which is a property of complex systems, onto a simple physical process. And finally, and perhaps more whimsically, really, I get the feeling sometimes when I'm talking to biologists that the problem is not a question of actual scientific disagreements with the bits and pieces of the theory. It's just the fact that it's very unfamiliar. All interdisciplinary work is by definition unfamiliar to one or another of the disciplines. When you're talking to biologists who don't really know much physics or chemistry or physical chemistry, you have a problem that half the time they don't actually understand what you're talking about. And so I summed it up in this famous sentence, I don't understand it, it must be wrong, which was uttered by a giant of 20th century biophysics, Max Delbruck, who was at Caltech. And apparently, I've never met Max Delbruck, but apparently a friend of mine who was there and knew him said that in seminars, he would, once in a while, he would stick his hand up and he would say in a strong German accent, I don't understand this, it must be wrong. And that sentence has become a sort of summing up of many reactions to new theories in general, not just mine.